So hello everyone, my name is Darla Saunders, and on behalf of the Atlantic Salmon Conservation Foundation and the Canadian Rivers Institute, I would like to welcome you to today's webinar. This series is made possible in part thanks to the contributions of the Government of Canada. For today's webinar, we're extremely pleased to be hosting Scott Roloson. Scott's PhD research has been conducted with the Canadian Rivers Institute and the University of Prince Edward Island and made possible with support from the Atlantic Salmon Federation and the PEI Wildlife Conservation Fund. Prior to graduate work, Scott studied fish community ecology in northern Alberta lakes and population dynamics of muskrats in PEI wetlands. His personal interests include hunting, fly fishing, and working on his family's horse farm. After the presentation, we will open the floor for a question and answer session. You'll have the option of either asking questions directly using your microphone, or you can type them in and we'll read them aloud to Scott. I will now turn the webinar over to Scott.
Thank you, Darla. I'll start by saying that it's a pleasure to be before you guys talking about my graduate research that I've conducted over the past several years at UPEI and all over the province. I must first start, as Darla mentioned, by thanking a couple key funding agencies. The Atlantic Salmon Federation's been behind this project from the start, not only financially but with technical support related to the acoustic tracking component of the study, and the PEI Wildlife Conservation Fund, which made possible a lot of the acoustic telemetry through their financial support. This project was really the brainchild of three key researchers that got the ball rolling in the onset. Fred Worski, he was at the ASF and now works with the Ocean Tracking Network. Todd Dupuis, who worked with the ASF, and Mike Vanden Heuvel, who's my project supervisor and the director of the Canadian Rivers Institute. Without these three people, this project never would have got off the ground. I'll start just by giving you an overview of today's lecture. First of all, we're going to introduce the rainbow trout. As many of you will be familiar with the rainbow trout, but introduce them as an invasive species, if we're going to use that nasty word. And then we'll talk about PEI as a study system. The key focus of my graduate research was actually the migratory work, and so that will be the bulk of the talk today, and then some other stuff that I've been working with in relation to competition with Atlantic salmon. So to begin, rainbow trout are native to the Pacific Rim. In that image in the top right corner, you can see the dashed area. That's the native range of rainbow trout, where they exist as steelhead or anadromous rainbow trout and resident rainbow trout. In the late 1870s, there was somewhat of a conquest as people were of the mindset that we could grow fish in captivity better than nature could provide them, and as we depleted our fisheries resources, we wanted to replace these. And in the 1870s, rainbow trout were found to possess suitable properties for propagation in captivity, as well as superior growth in captivity and in the wild, making them ideal for stocking initiatives. You see in the bottom right-hand corner, horse and carriage and milk crates were all that was necessary to transport rainbow trout eggs from one side of the country to the other, where they eventually made it to the eastern seaboard. When the water warmed up, they would throw on more ice cubes and carry on. Now you see the red area, rainbow trout are one of the most widely introduced fish species on Earth. You see their typical migratory range in the Pacific Rim out into the Pacific Ocean through around the Bering Sea. Some fundamental properties of the species in their native range. They exist with up to 5 other salmonid species, so competition is no to them. They're a spring spawner as opposed to a fall spawner, which our native salmonids on the East Coast are. Iteroparous, meaning they return to spawn more than one time, which is different than most of the salmonids on the West Coast, which are semelparous and die after spawning. And they may or may not go to sea. There's both a long-distance migrant type that goes across the Pacific Ocean, as well as a resident non-migratory phase. the current distribution of rainbow trout on Prince Edward Island is growing as we look harder and harder. You can see UPEI in 2001 formed this map that shows where they were established, but also the stocking history, this tiny blue text and arrows. It doesn't matter where and when and how many. What I want you to see from this is how many places, how ubiquitously they were introduced. And our previous knowledge of their distribution was based largely upon electrofishing surveys that went and looked for Atlantic salmon. So if they weren't in a salmon river, then there wasn't really anybody looking. So on the right-hand side, we picked and selected a few watersheds, and with the assistance of the provincial government, the yellow ones are places where we actually looked and found rainbow trout spawning, or not rainbow trout spawning rather, but evidence of successful reproduction in the form of either young of the year or 1-year-old juveniles. So the more we look, the more it appears that the southern edge of Prince Edward Island and the eastern side are well populated with rainbow trout. And I should mention before I go any further that more regionally there's also rainbow trout springing up in areas of Nova Scotia, New Brunswick, and Newfoundland, such as the Bredor Lakes, and I believe around the Antigonish Middle River in Nova Scotia, and Crooked Creek is one that comes to mind. People speak of having rainbow trout in New Brunswick, which I believe is in the Shepparton area. So they're starting to see rainbow trout in established populations across the region. And I guess PEI is a great place to study these rainbow trout because we have so many places where they have established, but also the urgency is that we want to know, hey, where are they going and what might the effects be on native Atlantic salmon. And you can see inside the circle, within a very short distance of PEI, some of the most iconic Atlantic salmon rivers in Canada and in North America. So it's a short migratory route to any of these should the rainbow trout be migratory and potentially invading other rivers in the region. And some of this work was brought about by the seminal research done by the Thibault lab, and you can see here they conducted a genetic profiling of rainbow trout that they were finding in the upper St. Lawrence estuary, and by and large it revealed that the invasion was fueled by mass stockings upstream in the Great Lakes region. However, the genetic profile that they conducted on 243 fish showed 3 fish that exhibited a genetic signature similar to the rivers that they tested on Prince Edward Island. Now, it should be interpreted with great caution because all of these fish at one point in time, and merely years to decades ago, not centuries, they all originated from domesticated stocks. So the question of whether they would have enough time to specially genetically distinguish from other groups, and did they all come from the same or different genetic origins? Those questions are underlying this research and whether or not PEI rainbow trout were actually migrating that far. But across the globe, it's popping up, they're popping up in other places. The picture here is what they call a unique Atlantic steelhead found in Argentina, and they grow as big or bigger than steelhead in the native range, but there's been very few cases on Earth where they've actually been found to establish a migratory seagoing life cycle phase. But Frosch in 2007 argued that one of the facilitators in further invasion would be the development of an anadromous life cycle phase, which would open the door to dispersal between adjacent waterways. So that was one of the first dangling yarns that brought about this study. The second one has to do with the more local interactions with Atlantic salmon. You can see pre-European settlement in the late 1700s, early 1800s. It is believed that Atlantic salmon existed in virtually all, between 80 and 100 waterways on Prince Edward Island. 2007 and 8, more disparaging times as they've been reduced to about 20 rivers. And Cairns went on to state that in 2012, all salmon rivers on the south side of PEI are below conservation requirements, and all of these rivers have rainbow trout. So those two dangling yarns were the impetus for this study, and from there, over the last 4 years, we've proceeded to look at the migratory patterns and ecology of the species of rainbow trout on PEI and their potential interactions. Now, I want to just talk for a few minutes about PEI. For those of you who aren't from here, you probably recognize the landscape and the character featured on this slide, and that's what the tourism industry of PEI would like to have you associate with, the pastoral landscape as depicted by L.M. Montgomery, and those of you who come here for you to see it for yourself. But there's a nastier side to the way we use our land base on PEI in such intense agricultural operations, and I call them the big three. Sedimentation is a huge issue on PEI, largely from exposed soil left unvegetated through agricultural practices, and also sedimentation from dirt roads. And this is as a scientist, it's very hard to get your finger on how much silt is coming in, and there's monitors and devices that can look at that now, but how is this affecting the aquatic ecosystems? That is a major question that we need to know much more about. The other two, eutrophication, again related to excess or intense agricultural practices, excess fertilizer entering the water table and eventually finding its way to estuaries on PEI. And one that PEI is frequently in the news for is pesticide-related fish kills, catastrophic die-offs that eliminate virtually all salmonids present in the stream. So I just want to sort of dwell on these for a minute because it really sets the tapestry upon which the interactions between Atlantic salmon and rainbow trout and brook trout occur. You see again, the pastoral landscape of productive coastal bay in the top left, and what is commonly left an open field, and then on the bottom three pictures you get an idea of how these can affect our aquatic ecosystems, with the sediment entering the woods, the sediment entering the stream. The middle picture is the Dunk River, and you can see two adjacent tributaries, one flowing in clear and one flowing in with much suspended sediment, and then further on the bottom right corner how it might look in the actual river and how it could potentially smother all aquatic life in the substrate. With respect to eutrophication, this is Pierre Gizzard figure from his thesis, and you can see that most of the nitrogen entering the Gulf of St. Lawrence, Northumberland Strait, is coming from Prince Edward Island. So we have some watersheds that are virtually turbocharged with nitrogen, and this has dramatic consequences for our estuaries where this nitrogen begins to be consumed. The anoxic events, and forgive me, that was supposed to be a map of PEI in the top white box, but what's important is you can see there's some serious physiochemical processes that go on once these fertilizer-induced plant blooms, once the vegetation begins to die off. There's, in this picture, sulfur-reducing bacteria. In the most extreme cases, actually turns the entire estuary milky white. And the third prong of the big three is fish kills on PEI, and since 1962, 45 to 50 fish kills have been caused by pesticides. These are recorded pesticide-related fish kills, meaning somebody actually has to find them in order for them to occur. How many occur without being found is another question up for debate. But in the sort of gray years or dark years between 94 and 2004, there was 22 fish kills. And of these fish kills, 16 have been attributed to a specific pesticide, and 10 of those involved azamphos-methyl or glutathione. Why I'm interested in this is there was a study that was published by Gormley at the University of PEI that showed after a fish kill that virtually all brook trout and Atlantic salmon were gone from the system, and rainbow trout were some of the only ones that were remaining. And you can see the LC50 for rainbow trout is almost 5 times higher than it is for brook trout, while salmon fall in the middle. And you can see here from the 1970s on the West River the absolute devastation that would occur to a river when you go in and remove all of the brood stock from the river. And to be truthful, the West River has never really recovered with its brook trout numbers to those former days. And again, I mean, these we like to think of these things in the bygone days, but this is 2011, and there's been more recent events. And this again, a canoe full of brook trout. So the issues that are before us in PEI rivers, rainbow trout rank low in the hierarchy of issues that are actually disparaging our fish populations, but nonetheless they are among the potential contributors to the demise or degradation of our native salmonids. So now getting on to my actual research, and from those early questions about the migration and the St. Lawrence, it prompted us to use acoustic telemetry to study the migratory patterns of rainbow trout in our rivers. So we selected 3 rivers with popular, well-known populations of migrating rainbow trout. And we eventually ended up teaming the acoustic telemetry approach with otolith microchemistry, which I'll elaborate further in a moment. But the goals here were to describe the timing of annual movements. Were the fish straying locally? Were they moving long distances? Or were they resident in the estuary? So just to give you a little further context for any of you who might not be fully involved in fish biology and fish tracking, the acoustic telemetry involves implantation of a surgical implantation of a transmitter with an external ID tag that enables identification of the fish should an angler catch it. And on the right is what's called a line scan from otolith microchemistry. So as the otolith inside of the it's called the ear bone inside of the base of the brain of the fish, and it grows as the fish grows. So as the fish grows and incorporates environmental chemicals into this bone, these tracers can be used to distinguish, for example, whether a fish was in freshwater or saltwater, how long they spent there, and so on. So with these 2 approaches, we were actually able to resolve more about the migratory patterns of the fish in our rivers. And I always found it amazing whenever I was out studying these fish that no matter what wind you think you might know what you're dealing with, or once you think you have it figured out, then they throw you for a loop. So the first year we thought we knew what they were going to do, and they did something totally opposite. But these fish are actually, and I use decisions with quotations because the decision is selected for by how many offspring these fish produce in the next generation. So if they find a successful migratory stage or phase, that will benefit the individual by the next generation, and therein forming a successful life cycle phase. So they're really faced with 3 choices, and we didn't know much about this when we started. So the 3 choices were: do they go nowhere, which is sort of their present situation, or the situation with, as they see it, with resource limitation, but to go far like they do on the West Coast, they really have no innate migratory instincts. So they're dealing with the high risk of mortality from predators in open waters, and the timing of migratory prey that may take, you know, hundreds or millennia of years in order for the migratory patterns to sync with the migratory patterns of prey. So in our estuaries and coastal waters, we have smelts and Gaspereaux and alewives and silversides that migrate through, but in the course of a few decades, it's virtually impossible for the migratory patterns to sync. And the third option, which became clear to us that it was the favored one, is go near. So they move down to productive coastal estuaries where the nutrient enrichment is fueling the coastal ecosystems, and their success in these environments is contingent upon the availability of prey. So one of the first in the first couple of years, we ran a counting fence on the Montague River, and all I want you to note here is like Atlantic salmon, like brook trout, there's a distinct phase in their life where they undergo a downstream migration and smoltify, which was really something to figure out early on, that okay, these fish are actually somewhat entrenched in an anadromous life cycle, because you can see that there's a succinct period of time and a relatively close size aggregation of fish that are traveling downstream within the same period of time each year. So there is an inherent smoltification that goes on in these rivers. So just to give you an overview of the acoustic telemetry, we studied the Montague, West, and Dunk, and each of the red dots is the placement of a telemetry mooring. So if you look at the West, that spans about 40 kilometers from the farthest most upstream to the farthest downstream, and with these telemetry arrays, we were able to delineate the migratory patterns of about 60 fish that were tagged. Some of those provided limited information, but some of those provided several 100,000 acoustic data points. And this may or may not work, and unfortunately the image is slightly cut off, but what I'm trying to show here is the movement patterns. That's supposed to be a fish, when really it's just the top half of a fish. I'll ask you to use your imaginations with me for a minute, but what this is showing is that, you know, the fish spent 98% of its time in a circle the size of less than half a kilometer in the river before returning upstream into fresh water later on in the season. So immediately we are, you know, we tag all these fish. We think, okay, they're going to go, you know, across the region, but really, no, they didn't. They remained within a very close proximity. And once we sort of started looking a little closer at the productivity in these areas, the enriched water fueling algal blooms actually produced copious numbers of these small gammarid amphipods, and like these fish were feeding on these amphipods, which drifted by on the tide. So they, with literally no energy expenditure, the fish would open their mouths, and this fish that was about a pound and, you know, the shape of a football, in its last meal had consumed over 60 of these amphipods. So we get an initial snapshot into the success of the life cycle phase, because they're able to reap the benefits of the estuaries, and this is probably one of the biggest fish that we've come across in the study, about 4 kilograms or 8 pounds, and anyway, from the original tracking, it becomes clear to capitalize on the buffet in our eutrophic estuaries. Again, you'll use your imagination to watch this half fish. This fish was the only fish we tagged on the Brudenell River, and as such, we nicknamed him Lone Wolf. and he was sort of an outlier in the first couple years, because instead of sitting in a very small area, he moved up and down the Brudenell River on a daily basis on what we presume to be foraging events, and then lo and behold moved into the Montague River, up into freshwater on the valley field. And so it was quite amazing, because this fish was actually captured again by my friend who was angling on the Montague River. Had no idea the significance of catching this fish, but you can see in 4 months the fish had almost grown 4.5 inches since it was tagged in May and recaptured again in September. So not only are they gaining, not only are they growing quickly in limited areas, but they're also growing quickly when they're on the move. And who knows why this fish actually came over to the valley field Montague system. But what is quite impressive is that this fish on December 29th turned around and returned to the home pool where it initially tagged on the Brudenell River. It did this about 17 kilometers in under 1 day, which again, just when you think you know something about it, questions my understanding of the osmoregulatory, the physiological capacity of one of these fish to go from pure fresh to pure salt back to pure fresh within a 24-hour period. So it's quite clear that these are very hardy or resilient animals. So to look at the tracking in a different light, just to sort of do a better job of summarizing it, this is a average daily position, each point. So you can see the line 0 represents what we designated as the head of tide. So river kilometer 0 is right where the fresh and salt water begin to mix, but it is more fresh than it is salt influenced. So you can see in the first one, which we would classify as a marine fish, he moved about 20 kilometers downstream, and then throughout the summer months registered scattered isolated detections, and then through the month of March in 2013 actually moved about 20 kilometers upstream to spawn. But that fish is the exception. The second fish on this slide is the norm, and you can see that there's a distinct period during the summer of residency where this fish is staging in the cold water plume of the river nearby to the salty marine waters, but where there's refuge from temperature, high temperatures in the summer. And the bulk of the movements of this fish actually occur after summer's over, September, and then October and November, and even into December. You see that fish is on the move every day, and it's always, you always want to hope that your research is going to be grounded or accepted in the public and management decisions. And it was pretty amazing that based upon this information, we know that these fish are on the go throughout the fall months. There's been 60 days of rainbow trout fishing added to the end of the season since this research was conducted. So managers are accepting this and moving towards managing our resource instead of considering it a bane and only a bane. Now I want to talk about some of the otolith microchemistry. So if you look along the bottom axis from 0 to 2000, that actually represents distance across the otolith, but it also represents time in the fish's life. And the green line corresponds has 3 explicit migrations into marine waters, whereas the bottom fish as indicated by barium, the purple line, shows more of an occupation of the transitory zone like the resident fish does. So it doesn't clearly register the marine signature. It shows a mild marine signature, but this is one of those estuarine resident fish that is moving up and down the salinity gradient as conditions permit. And we got really curious about this after sort of seeing it unfold before us. And in 2014, a colleague, Mike Coffin, and myself set up some oxygen temperature loggers, coupled them with our receiver arrays, and used that to delineate real-time oxygen temperature and fish movement patterns, and then in an effort to sort of see what was really driving these movements. And you can see, point 0 is 1 kilometer from freshwater, which would be near the road, 110, the designated freshwater, and then gradually it becomes more salt influenced as you get out to kilometer 5.9. So just bear in mind where these points are as we look at the next slide. The dissolved oxygen and temperature recordings show that at kilometer 5.9, the green, and at 3.9, the off yellow, that there's daily fluctuations, and through June, July, and August, that these sites actually go to 0 oxygen. And in the background there, you can see 1 and 2.5 remain much more stable. And similarly with temperature, the months of July and August actually see daily temperatures going up into the region of 25 degrees. So it explains a little bit on the next slide here about those estuarine residence periods throughout the months of June, July, and August. And it's particularly clear from the telemetry. And this research has provided us with a fascinating opportunity to compare and contrast acoustic telemetry and the fine real-time daily resolution and autolith microchemistry, which is more of a delayed incorporation as the fish is moving from environments. It gives you more of a summary record. So we can see here that the autolith microchemistry isn't providing the same degree of resolution as the acoustic telemetry, but on the flip side of that coin, the autolith microchemistry can give you a profile over the entire life of the fish, whereas the tag lifespan restricts your acoustic telemetry findings. Another powerful insight that's given to us by autolith microchemistry is the contribution of an anadromous mother to the next generation. So if you watch the green line, starting from the beginning of this fish's life from the West River, you can see it starts with a strontium spike. That is a reflection of the young fish relying and assimilating the maternal embryonic sac as it's coming out of the gravel and absorbing the maternal nutrients before it becomes a fully independent and feeding fish. So this shows us that the maternal anadromous mothers are contributing, shows us what they are contributing to the next generation. And on the dunk, out of the 32 fish we looked at, it was 9.3%, 28 for the Montague, and 54 for the West. On the dunk, that may be obscured, however, because some of these fish, in light of the terrible water quality conditions, some of these fish never actually make legitimate salt water. In fact, they spend most of their migratory period in the low salinity transition where they're finding refuge from the poor water quality. Okay, so now what the title kind of alluded to was the interactions with Atlantic salmon, which is an independent chapter of my study altogether, and something that I've just racked my brain, decipher the way, because it's based upon such complicated interactions of two life cycles that are very different but also very similar. So just to sort of break the life cycle down into components, Atlantic salmon are obviously obligate migrators. They smoltify after 1, 2 to 3 years and go to sea. Rainbow trout are much more facultative. They may or may not. They don't have to, but they will. Again, though, they rear in stream for 2 or 3 years. The key difference between these two, I think, is the fall versus spring spawning, which may have dire consequences for one versus the other. Sort of elaborate on that again in a moment. The rainbow trout and juvenile and Atlantic salmon juveniles are both regarded as competitors, and it's often perceived that rainbows are dominant in this competition, but the competition for high gradient riffle habitat is believed to be the key competitive arena where rainbow trout are sort of pushing Atlantic salmon out. Just to sort of put for some food for thought, I took the Water Office Environment Canada hydrograph for the West River for a whole year and just highlighted the theoretical incubation period for Atlantic salmon and rainbow trout. And you can see that rainbow trout, A, is a much shorter incubation period, but what I really wonder is what about the added risk of having your eggs incubate over a greater period of time? So during the incubation period of Atlantic salmon, you see between 10 and 20 flood events or water level peaks, and some of these are the high water freshet for the season, which can result in entire substrate turnover, whereas rainbow trout tend to come in and spawn after the spring freshet and water levels are continuously subsiding. But what became clear to us early on, and it was from watershed groups spawning records, is that rainbow trout may be coming in and spawning in the same areas as Atlantic salmon before Atlantic salmon emerged. That's one of the two key potential axes of competition is this exclusion or wholesale devastation of the salmon reproductive event by excavating their reds while they incubate. And I had to, one of those things that I really wanted to get to the bottom of, and I had it in my mind that we could take 3 rivers and survey where the salmon spawned, and then go back again in the spring and find where the rainbow trout spawned, and if there was overlap, we could deploy these drift nets and check them periodically. But it became clear after trying to do the spawning surveys that the odds of finding the exact place in the river where a rainbow trout had spawned over an Atlantic salmon was astronomically low. First of all, you're dealing with populations that are maybe in dozens or hundreds of individuals, and it was sort of apparent after doing the surveys that they were actually spawning in different areas of the river. Rainbow trout were going up to 20 kilometers upstream, whereas the salmon were isolated to the main stem. So this component of the study, while very beneficial in determining where the spawning occurred, it was fruitless in terms of actually finding that magic bullet. Yes, rainbow trout are digging up Atlantic salmon, and therefore we should wage war on rainbow trout. But we came up with another approach that looked at the juvenile life cycle stage. We looked at the Cross River, which has only Atlantic salmon and brook trout, and Pisgah and West River that have both Atlantic salmon, rainbow trout, and brook trout. So we did a sort of a two-pronged approach with electrofishing surveys, and we wanted to cover the longitudinal gradient of the river. So going from the mouth, the first suitable juvenile habitat, to the tributaries, to the very headwaters, strategically place sites on each of these areas on each of these rivers. So from the electrofishing surveys, we covered 65 sites, and we actually looked at 5 different rivers with the electrofishing surveys, but ended up only being able to make it back to about 45 sites for the habitat surveys. But so in the habitat surveys, we went back and surveyed slope, and then some point measurements of velocity and discharge, which allowed us to calculate some more energy-related hydrological parameters with the system, such as bankful, discharge, and bed in motion parameters at present water level and bankful, and substrate size distribution. So we measured 100 rocks at each of these sites, and you see the rock there with the X, Y, and Z. Between 45 sites with 100 rocks at each site, that includes 13,500 rock measurements. Yes, I got tired of measuring rocks. Sorry. So this is what we end up with, looking at the relative abundance of the juveniles that we encountered. And the main stem, you can see that it's largely salmon dominated, WM5, there's about 30% rainbow trout. And then we go upstream a few kilometers to the Quinns Brook tributary, and you can see there's no salmon and between 80 and 90% rainbow trout until you get to the headwaters where brook trout become dominant. And similarly, brook trout became dominant in the headwaters of the other tributary, but there was very few salmon and some rainbow trout, but not many in the tributary. I should mention that this is sort of the analysis we've done on this is very preliminary and ongoing, and I'm sure that it won't be our final analysis, but what we've done is thrown it all into a principal components analysis to see if we can tease out some of the variables that are driving the occupation of either Atlantic salmon or rainbow trout in these sites. So we're sort of imposing the habitat variables on the fish community information, and just it looks kind of messy for now, but I'll have a look at it here, and all I want you guys to take from this is that you can see that on the right, the 2 areas that are Atlantic salmon dominated, this is where Atlantic salmon were the most abundant of all fish species in the community, and the drivers that seem to push those are large cobble and small cobble. So the presence of rocks that are over 65 millimeters, say, would be small cobble, so big enough for the salmon juveniles to hide under, but also width, stream width, and hydraulic radius, so the area of stream bottom, which incorporates width and depth. And then off to the side in the red circle is the area of Quinns Brook that was dominated by rainbow trout. And these are high slope areas where the bed mobility is high and so on. And then the sort of smattering of other sites that are outside of the circle is just a reflection of the sites where brook trout were dominant, and they all sort of group together on the side. So again, this stuff is very preliminary, this analysis, and we'll be going through it again and again, but we just wanted to be able to share with you some of the things that we're coming across, and our initial findings suggest that the presence of suitable substrate as opposed to the presence of rainbow trout is what dictates where we will find salmon. And you can see that because all of the sites from the Cross River that have no rainbow trout, they match up perfectly with many of the sites from the Pisgah and from the west where the substrate size is suitable. So that's all I wanted you guys to take from that. So just a quick conclusion here. Salmon juvenile distribution appears to be related to substrate suitability. With respect to migration, the rainbows show an incredible flex confronted with adverse and changing water conditions, and this flexibility may be favorable when and if they disperse into new waterways. And the sort of upshot of all this is that now rainbow trout have become an important angling subsidy, and they must be considered as such when we're looking at their benefits and detriments to both angling and ecosystem integrity on PEI. And there's a ripe male rainbow trout with his spawning regalia on in the spring in the West River, ready to go, and I would be happy to answer or field any questions you may have.
Thank you, Scott. That was an excellent presentation, really informative. So as Scott said, we'll now open the question and answer session. So just to recap, to ask a question, you can use your webinar control panel, which is the little gray box you'll be seeing in the upper right-hand corner of your screen. If the box is minimized, just hit the orange arrow and that will enlarge it. So you can either raise your hand, figuratively speaking, by pressing the yellow hand icon with the green arrow, and we can unmute you and you can ask your question to Scott directly, or you can type in your question on the control panel and we'll read it aloud for you. And so I think we've got one question here already for you, Scott. So when you were showing your animation, of movement. The question is, how are you tracking the movements of these tagged fish? Were the blue pins on the map shown receivers, or are they showing tagged fish? And that's from Jessica Gideon.
Okay, Jessica, sorry if that wasn't clear. Just go back to it. Yes, to answer your question, the blue points are fixed telemetry arrays, so it's much like a scanner in the grocery aisle. When the item goes by, the fish, it records what item it is and ID and time and date. So there's a software package that houses all of these detections, and then from that you can construct where and when these fish were at given points in time.
Thank you. So the next question comes from Fred Worski. He asks, some very small rainbows, less than 6 centimeters, move downstream to the estuarine zone. Do they go to the same places as the larger fish?
Hi, Fred. What I think is happening is it's just sort of a consequential thing. They just end up coming downstream. There's a few, maybe 200 meters of holding pools below the fishway, and as the summer goes on and the young of the year fish are actually on the go, we'll start to find more and more of these fish coming down, and I think it's just a fluke that they end up going downstream.
Thank you. So the next question comes in from Rick Simpson. Rick says, wow, thank you. What will your likely future research focus on?
Thanks, Rick, for your kind words. I am fascinated with migration, and pretty much that's it. So anything, I mean, obviously there's a lot of ecology in what I'm doing and working with here, but I find that migration in anything, and I mean it's not only fish migration, there's all sorts of other great migrations that go on, but that's the thing that can make the hair stand up on my neck in any day of the week is the powers at work in watching and finding out more about these animals that navigate, you know, across jurisdictional boundaries.
The next question comes from Samuel Guerin. He asks, did you do the 100 rock count in different habitat types or just in riffles?
The 100 rocks were measured within the discrete boundaries of our study sites where we electrofished. So I would flag the bottom and the top of the site and measure the rocks throughout. And there's no perfect way of doing that, and ideally I would have, you know, sampled both rocks, riffles, and pools, but it was sort of limited to the defined area of the sites.
The next question comes from Peter Cronin. He asks, what is the current PEI rainbow trout stocking policy and their rainbow trout aquaculture policy, private fish ponds?
There are ponds that are still ongoing on PEI. One sort of a catch and keep pond and that is still ongoing. There's another one on the West River that grows rainbow trout to fingerlings and sends them off for sea cage growth. It's still permitted. I don't know a lot about the specific regulations that they deal with, but rainbow trout stocking into open waterways has been eliminated on PEI, so there's no more stocking of any rainbow trout into open waterways.
Okay, our next question comes from Chris Elvich. Chris asks, toward the end you showed a strong longitudinal distribution of the three species. Could upstream consumption of drift forage by rainbow trout be driving sympatric rainbow populations downwards?
Sorry, so I'm not entirely clear on that question, Chris. I guess I'll just throw an answer out there. There's very little that can deplete drift forage in PEI rivers because they are amazingly productive systems because of all the groundwater nitrogen that is flowing in. So what's likely driving the upstream occupation of brook trout is the cold water refugia, and there's still certainly a lot that we don't know, because it appears that rainbow trout do well in sort of the intermediate tributary areas, but once you go to the headwaters, which is commonly found in the literature, brook trout do dominate. And so I'm not sure if that answers your question or not, but I mean, there's still a lot we don't know.
Thank you. Chris, if that doesn't answer your question, please type in again. Our next question comes from Catherine Collette. Did any otolith or other data suggest that any of the fish you studied originated as aquaculture escapees?
That's a great question, Catherine. I scratched my brain. I actually did the otolith microchemistry in New Zealand, and I found two fish on the Montague River that were big, like we'll say 5 pounds. And I could not find a marine signature at all. And it was like 2 months later, whenever the epiphany struck me that these fish, my explanation for it was that these fish originated in the catch and keep fish pond that is located in the very headwaters, and then sort of spins this whole idea of what that fish pond may be contributing to the stock, be it in individuals that might reproduce or actual angling fish that are angled, because there was a fish in that same river this year that was caught about 29 inches in the spring, and that's like 4 inches bigger, 3 inches bigger than the biggest wild ones that we encounter. So it is a very real possibility that in those very isolated cases that escapees are mixing with the wild population.
Excellent, thank you. So I think that's the end of our questions.